Okay, let's pick up where we left off. So we had previously covered how the structure of DNA, how DNA is replicated, and we talked about sort of all the sort of proteins and things like that involved with DNA replication. But let's talk about what happens when DNA gets damaged, right? Either due to a mishap during replication or due to some external forces. So, as you recall, any changes to DNA is simply what we define as a mutation. And mutations typically um, are generally defined as being spontaneous. Um, however, there's certain things via direct exposure that can certainly increase the odds of a mutation occurring. Now, what are the sort of the main types of mutations we're going to focus on? Well, the first is spontaneous, right? Mishaps essentially during replication, and these form what we call tautomers, and we'll show you what that looks like in just a moment. We also see slippages during replication. So polymerase essentially slips, makes an error, not caught. And then we also have spontaneous damage to individual bases. So it's not super duper common, but it's also not uncommon enough given the size of a genome. But let's sort of take a peek at what we're looking at here, right? So we have AT, C, and G at the top here. Now, where we have sort of these tautomers forming, where we have T binding with G, we see that here twice, and C binding with A, as we see down here twice. So the idea here is that we have this essentially this rare type of base pairing forming clearly a mismatch in DNA, right? Now, how does this form? Well, think about it, right? We have hydrogen bondings, right? That's what holds these things together. And the reality is an N and an H or an O and an H can form any hydrogen bond. It doesn't really matter so much, right? And so these arise simply just because of how DNA is typically held together. But you can imagine essentially swapping out a C for a G or a T for an A or something like that can have some pretty serious side effects, in particular if you essentially change something that's happening inside the protein structure, or I should say the protein's primary sequence. Now, another example we see is where we see errors in replication during regions of highly repetitive DNA. So as we discussed, the centromere and the telomeres are both areas of highly repetitive DNA. And so one of the common things we see is what we call trinucleotide repeats, which are actually susceptible to what we call strand slippage. And the idea here is with strand slippage, DNA polymerase replicates a short section of DNA essentially twice, right? And so we have essentially a doubling of a piece of DNA. Now, this might not seem like a huge deal, and maybe it's not, right? There's large areas of eukaryotic genomes that are non-coding, so sometimes a sort of a repeat is not such a big deal. But there's some pretty well characterized human diseases, which is defined as trinucleotide repeat disorders that do involve the accumulation of these repeats over time. But let's take a peek at what we're looking at here. We have DNA replication, we have the strand separating, which then we have the beginning of replication. As you see, we have DNA polymerase binding here. Now, what ends up happening is we have this slippage here, um, and we have this repeat, right? CAG, and so on and so forth, right? And so we have this slippage, um, and it causes essentially the strand to detach and rearrange. And so we have this little hairpin loop, and this little hairpin loop is recognized as something that's bad. And the next time these things happen, next time this sort of goes to be replicated, this little hairpin, being extra pieces of DNA, will actually be replicated, elongating the original strand that we're talking about here. So this is what we're looking at when we're looking at strand slippage. And so here's just a couple examples of some of these trinucleotide repeat disorders. Many you might have heard of, including Huntington's disease, Fragile X syndrome, or maybe you haven't heard of any of these. But here what they are. Here's the repeat sequences. Here's how many normal there are, and here's how many are there in the disease, and here's the primary symptom. So lots of sort of interesting diseases, very deadly diseases, of course, that arise with these slippages. Now, another thing we commonly see is depurination and deamination. Now, this happens spontaneously, and it's not so much a DNA slippage or thing or alteration of bases in the way we've been thinking about thus far, but we're mostly thinking about the loss of a purine base for depurination, or for deamination, we're talking about the removal of the amino group. And so depurinations are pretty common. As this sort of tells you, a human cell can actually undergo thousands of depurinations every day and about 100 deaminations. So it's pretty common. So let's take a peek here. So we have the backbone strand of the DNA here. We have guanine. It gets depurinated. We lose that DNA base. Now we're missing the guanine here. Now we have nothing to essentially bind to the other side. So the DNA potentially become more fragile, improper replication, and things like that. So that's one. Now, deamination is the loss of an amino group. So we're losing this amino group right here, and we're consuming water in the process. So we're essentially replacing the amino group as well as this double bond with water, right? OH. Um, and the other H obviously is tacked on here, right? Because we go to NH2 to NH3. So we consume water. And what we actually do here, and this is kind of a cool thing to think about, is we actually go from cytosine to uracil. Now, as you recall, uracil is an RNA base. And so we have a spontaneous generation via deamination, putting uracil, and this isn't the only outcome, of course, but in this case, it's putting uracil in DNA. So an RNA base in DNA. So clearly not not ideal here. Now, the one you probably know most commonly is the idea of mutagens, right? These are chemicals or some sort of agent, environmental in nature, that causes damage. And there are two broad categories: chemicals and radiation. So let's take a peek at that. Now, one of the common things we see with our chemical mutagens is actually what we call base analogs, where they actually will be mutated into something that resembles our nitrogenous bases and are actually directly incorporated into DNA. We also have what are called base modifying agents, which actually can simply react chemically with DNA and the bases in DNA to alter their structures, forming what we call DNA adducts. And I'll have a picture here just to sort of show you what that looks like in a moment. And then we have what are called intercalating agents, which actually essentially insert themselves between adjacent bases, distorting DNA structure. Now, for our base analogs, they are very structurally similar to one of the DNA molecules. They can be incorporated. Now, a common example we see of this is actually BRDU, or 5-bromodexyuridine, which actually has very similar base pairing properties to thymine. However, when it's incorporated into DNA, the DNA is replicated, they actually incorporate an A into the brand new strand. So it's similar to thymine. So we replace thymine with BRDU, but when we go to replicate it, it recognizes BRDU is actually adenine. So it actually puts it, when it's replicating that DNA, it puts an adenine instead of a thymine. So we have a single nucleotide polymorphism or a single nucleotide base change. Now, there are many, many types of base modifying agents, so changing a T to a C or something like that, and they chemically modify these bases. So thus there is a mismatch in the next cycle of replication. Now, common examples of this including ethyl methanosulfonate, or EMS, which actually will add ethyl groups to bases. And we also have another compound called nitrosoguanidine, which will actually add methyl groups to our DNA bases. Now, as we talked about with DNA methylation a couple slides ago, that can also affect how DNA is accessed by DNA polymerase. So it not only affects the bases, but it also affects transcription as well. Now, nitrous acid, it's a pretty common thing. As a result of deamination, this can actually base modify our things. And there's lots of other sort of common compounds out there that will essentially modify or essentially replace what we're seeing here. So for instance, we can see a base here, example here, where we have DNA, we have guanine, we have the DNA backbone here, and we have an example of alpha toxin B1, which will bind directly to this guy, working its way in here, distorting the structure of DNA. So there are lots of these, what we call intercalating agents. They're simply altering the shape of the backbone of our DNA. And the reality is once they are repaired, so once you try to repair something such as this, what it actually does is it causes oftentimes single nucleotide polymorphism. So for instance, in lab, we worked with when we do gel electrophoresis on our halobacterium lab, we'll use a compound similar to ethidium bromide, which actually intercalates itself into DNA. And ethidium bromide, like the one we'll use in lab, is fluorescent. So when we shine a UV light on it, because it's intercalated itself into DNA, it allows us to visualize DNA. It's kind of a cool thing. So next up is actually radiation. So those are all chemical means. Now, radiation is pretty common. Now, radiation is simply referring to electromagnetic radiation, right? Stuff that comes from essentially the world of light, right? X-rays, gamma rays, and so on and so forth. So lots of things that encompass this. Now, what do they do? Well, essentially what's happening here is if we have thymine here, we have ultraviolet radiation, so essentially what causes skin cancer, right, from the sun, of course. One of the common things that happens because of this radiation, it causes what we call thymine dimer, where thymine will actually bind to the neighboring thymine, thus not binding here with its adenines as it should. Now, what does this do? Well, the reality is this type of binding weakens DNA, so DNA can start to break, things like that. And oftentimes when these are repaired, they're oftentimes just kind of excised and you lose a chunk of piece of DNA, though it does vary um, depending on the organism involved, of course. So those are our types of mutagens. Now, how do we fix these things, right? What are our DNA repair systems? Well, the cool thing is there's lots of them. There's lots of types of DNA system repair systems out there. So the first one we'll discuss is actually light-dependent repair. So these actually fix pyrimidine dimers, so or thymine dimers. That's what we're thinking of here. And this is what we call photoactive repair. Now, it does depend on an enzyme called photolyase, ACE being an enzyme, photo being light. So it's an enzyme that works with light to do its activity. And so that's one. Now, another common thing we see is simply called base excision repair, where essentially um, it corrects a single damaged base. So for example, if you have a deaminated base, they're detected by an enzyme called DNA glycosylases, which actually will remove the base by cutting the bond between the base and the sugar, and then replacing it with the correct DNA base based upon its partner across the DNA strand. So we can sort of look at an example of this. We have a modified base where we have GC and then anything X, right? Anything but C right here. So it's going to be T or A or even another G. What our DNA mismatch repair does is it uses this enzyme DNA glycosylase to excise X. Then we have a free site where there's essentially an A and P, so a phosphorylated site, of course. Now there's another enzyme that comes in, excises that bad backbone of the sugar. DNA polymerase then comes in, recognizes the base across from it, tacks on the right base, thus fixing the problem. So a multi-enzyme solution here between glycosylase as well as exonuclease, followed by our friend DNA polymerase. And then the final step here that I didn't make mention of, but it's not super duper important, is that the final sort of ligating step here is done by our friend DNA ligase. So next up, we have what we call the nucleotide excision repair, or NUR. Now, this just uses proteins that actually detect distortions in the helix, so the 3D structures, and in recruit NER endonuclease that essentially cuts the backbone on either side of the lesion, or essentially the damage to the DNA. So let's take a peek at what we have here. We have UV damaged DNA. We have a thymine dimer. Remember, thymine dimers are a result of um, damage from ultraviolet light, typically distorting the overall structure of the DNA, weakening the structure of the DNA. Now, we have a complex which is called the UVR-AB complex. You don't need to remember what it is, but that's just what it's called. It binds to the opposite side of the dimer. Then one subunit of that protein departs, catalyzing a cut here. So 3' to 5' direction, leaving us an leaving a cut and a cut. So you see the cut here and the cut here. Then our then our DNA polymerase does come in, recognizing after the cut has been excised, synthesizing a brand new strand based upon the template, thus fixing the DNA. And then finally, DNA ligase comes and restores the intact piece. So lots of similarities to what we see here to what we see here, right? Recognizing the issue, cutting it out, then fixing it with polymerase, then ligating it back together with DNA ligase. So really similar mechanisms across these systems. Now, one of the things we actually commonly see is the role of methylation after DNA after methylation in mismatch repair. Now, as we talked about, methylation is an important part for turning on and turning off genes for transcription. However, methylation doesn't simply occur directly after DNA replication. So if that's the case, if methylation takes some time after a cell replicates its DNA, a mismatch repair system has to be able to distinguish the original DNA, which is methylated, from the newly made strand that is unmethylated, and thus the incorrect nucleotide in the new strand must be excised and replaced. So we can see what we have here. We have a mismatch here of GT. You see we have a methylated thing here, methylated thing here. Then we have an enzyme called a MUT-S. It recognizes the mismatch pair. Then we have another enzyme called MUT-H, which recognizes the methylated parent strand and nicks the daughter strand, so cutting this part. And then eventually the new strand is removed and replaced between the nick and the mismatch, allowing us to repair this in the presence of methylation, preserving that methylation on the parental strand. And so this is what we went through, just so you have it. Now, there is sort of this final thing we'll make mention of for the handling of damaged DNA, and this is what happens when sometimes things are just too damaged. What happens when things are too damaged? What do we do, right? How do we save ourselves when things are too damaged? Well, the reality is we can actually look to our friend E. coli and what we call its SOS system to actually figure this out. Now, SOS simply refers to like that old sailor's adage of save our souls, right? You would write SOS if you were stranded on a desert island or something like that. So that's where that comes from. And so E. coli actually has an SOS system to deal with it. So let's talk about sort of the role here and what happens with E. coli. So essentially the DNA in front of the stalled DNA polymerase, so it recognizes a really bad sort of damaged piece of DNA. The DNA polymerase doesn't know what to do with it, so it stalls. And then a protein called REC-A will actually join all those single-stranded binding proteins on the single strand. REC-A then itself leads to the expression of a special DNA polymerase, which we call bypass polymerases, which will then continue DNA replication despite the damage. And once the damage is passed, DNA polymerase 3, so just a different type of DNA polymerase, can actually resume the replication. So it's kind of a cool thing. The E. coli recognizes bad stuff and can essentially fix its issue overall. So that's E. coli. Now, in eukaryotes, we have what's called translesion synthesis. Now, this type of synthesis can actually produce brand new strands from the damage has been eliminated. So for instance, we have a polymerase called polymerase N. I forget what that Greek letter is called off the top of my head. You can Google it if you want to know. But polymerase N can actually catalyze the brand new synthesis across a thymine dimer, actually correctly inserting 2 new adenines in the brand new strand. So we have a couple extra slides here for you about recombination and transposons. Really, really fascinating area of biology, in my opinion, but sort of beyond the scope of what we're going to be covering in class. So I just want to leave that here for you so you can take a peek at transposon, because again, I think it's pretty cool. So, all right, there we go. So that's the end of that. Now this brings us to part part 3, where we're actually physically looking at transcription as a process. So how do we express our genes? So the production of messenger RNA or any type of RNA from DNA is what we call transcription. That should sound pretty familiar. Now you remember we have an overall flow to genetic information. This flow of DNA to RNA to protein is what we call the central dogma of molecular biology. So you want to keep that name, at least the central dogma, in your brain, because I'll refer back to it throughout this talk, and I'll refer back to it in other lectures as well. So the central dogma simply refers to that overall flow of information, DNA to RNA, then to protein. And then we have transcription, which is part of it. And that's where we essentially create RNA from DNA. And then we have translation, where we make protein from DNA as well. I'm sorry, protein from RNA. Oh, so many things. So we do have a couple special types of RNA. RNA isn't just a one catch-all thing. So you guys probably recall we have messenger RNA or mRNA. This is the RNA that's directly translated into protein. We have ribosomal RNA, which is an integral part of the ribosome. So it's part of the structure of the ribosome, helps with recognition and translation and things like that. And then we have transfer RNA or tRNA, which are molecules that actually serve as intermediates. They bring amino acids to the ribosome. Then we have rRNA, which are then folded, and they're actually interlaced into the ribosome, allowing the ribosome to function. And then messenger RNA, again, being the part that grows directly into translation for the production of proteins. So where translation and transcription occur does vary, right? Bacteria don't have a nucleus, so their transcription occurs and translation occurs both in the same spot, which is simply the cytoplasm. Whereas in eukaryotes, those differ a little bit. For eukaryotes, our transcription starts in the nucleus. After our primary, or what we call pre-mRNA, is produced, we have what's called RNA processing to produce a mature mRNA, which is then exported from the nucleus to proteins where translation can occur. But what you'll notice for our prokaryotes, it occurs right in the cytoplasm, right on where the DNA is. And what you'll notice is that there is this separation of space for eukaryotes. There's actually no separation of physical space here for prokaryotes. So once they produce mRNA, while it's still being synthesized off the DNA strand, ribosomes can directly bind and then begin translating right away. Kind of a wacky and wild thing they do. Now, this is actually what we call polysomes. And so you can see an example of this here, where we have transcriptional direction going here on our DNA. We're producing a longer and longer piece of um, RNA here. And you notice that as the RNA is being produced right off the backbone of DNA here, we actually have multiple ribosomes binding, doing multiple syntheses of proteins at the same time transcription is occurring. Now, you might ask yourself, why are polyribosomes or polysomes a thing? Well, the reality is, if you're a microscopic organism, you need to respond very, very fast. And being able to, and having no separation of transcription and translation means you can act really fast. But if you have a separation of transcription, as we see in in eukaryotes, there's a bit of a time lag. So just kind of a cool thing that we see here for our separation of space for our, space and time for our prokaryotes and eukaryotes. So just a couple little bit of extra information about retroviruses. Retroviruses are really cool. This is where HIV falls. So these are viruses that have RNA. They have a special enzyme called reverse transcriptase. So definitely take a peek at the slides if you're interested in HIV or viruses in general. But a little bit beyond the point of what we're covering in today's class. But again, still think it's pretty dang interesting. HIV is clearly one of the most important diseases of the past 20 to 30 years. So certainly bears take a peek if you have the time, of course. So we have some more extra slides for you here about the history and discovery of the genetic code. If you don't know about this, if you don't know about Beadle and Tatum, if you don't know about some of these other peoples, including Pauling, Ingram, so on and so forth, definitely take a peek at these slides about the history of the genetic code and discovery. Really, really cool idea, because we do take it for granted, right? We do take the idea that A binds with T, G binds with C, that every codon or 3 bases of DNA or RNA corresponds perfectly to a amino acid. But back in the day, in this little history lesson you could take a peek at, that wasn't always the case. So as we talked about, genes are the parts of and the functional aspects of DNA that encode for proteins. These are the things that are expressed. Now, as you probably know, there are 4 DNA bases, A, T, C, and G, which then specify 20 amino acids. Now, we do know this is a triplet code. There was a lot of debate throughout history that it was a doublet code, just as a note. That's why I put that there. But it is a triplet code, which means that for each of those 20 amino acids, there is a set of triplet codes. Now, if you're doing your math, if I have 4 DNA bases and it's a triplet code, 4 times 4 times 4 should give me 64. However, you'll notice that there's not 64 amino acids. There's only 20. So what does this mean? Well, this tells us a little bit about the genetic code. And the genetic code is what we call redundant, in the sense that there are multiple codons, typically speaking, for each amino acid. So if I had CCC and CCG, they probably code for the same amino acid. And actually, the way we figured this out was actually by frame-shift mutations. Now, when we insert or delete a nucleotide, we call these indel mutations, which actually causes the entire sequence to be read out of phase. This is what we call a frame-shift mutation. And so we actually can sort of look at this. So we have just a sentence here, right? Our big red dog bit the old man. I know it's kind of sad, but that's the sentence I pulled from this figure. And then we have a frame-shift mutation where we insert a base here, or delete a base here. Thus, we change the reading frame. So instead of our big red dog, we have our so on and so forth. Conversely, if we deleted a base, we go our and so on and so forth. So that's where we have a single mutant plus or minus. To a changing of your sentence. And that's really all a protein is, right? It's a sentence of amino acids. So the genetic code, as I mentioned, is degenerate, right? Meaning that there is multiple amino acids that correspond to what we see. In addition, it is also what we call non-overlapping, meaning that the reading frame advances 3 nucleotides at a time. So we go from our big red dog, so on and so forth, not our, and then B red dog, so on and so forth. So it always advances 3 at a time. So in this case, we have AUG, GGC, and UCC. That always goes methionine, glycine, and serine. We don't do any other way of reading that. It's always in chunks of 3. However, there's our code is non-overlapping. So what that tells us is that there's no overlap in between these bases, right? There's no, I use AUG only for methionine. I don't use AUG and then GGG for glycine, right? Only 3 DNA bases or RNA bases correspond for each thing. Now, the final thing I'll make mention of here is you'll see there's 6 lines, oh, I'm sorry, 7 lines here. And these actually correspond to a level of reading frames, but we won't really get to that. So the goal here is to take DNA, make a copy of it, and that's what we call messenger RNA. And that messenger RNA allows us for us to synthesize a brand new spanking protein. So in mRNA synthesis, a single DNA strand is copied. And we call this DNA strand the template strand. And the other strand is actually what we call the coding strand because it's actually similar to the mRNA sequence. And as you recall, in mRNA, uracil is substituted instead of thymine. So for instance here, if we have the coding strand on the top and the template strand on the bottom, right, 5' to 3', the resulting mRNA produces AUG, GGC, and UCC. Now you'll notice that's actually identical to the coding strand at the top. So when you break your DNA open, you copy the bottom strand, the template strand, but you're actually making an exact copy of the coding strand, right? Because that's what complementary base pairing tells us. Now, once you have your mRNA, it has polarity, right? Directionality, 5' to 3'. And we actually read that same 3-letter code, AUG, methionine, GGC, glycine, and UCC as serine as well. So you'll notice that the mRNA is identical to the coding strand, right? And that coding strand dictates the protein. But again, we use the template strand as a means to make a copy. Now, the question I could ask you, if we were in class together, of course, but we are not, is why don't we just use DNA? Well, the reality is DNA is too important to potentially be damaged, right? So we make a copy of it with messenger RNA to essentially save DNA, essentially allowing us to essentially preserve our DNA, our instructions, right? Because that's what DNA is good for. Now, we have, as I mentioned, there are 64 potential codon possibilities, right? 3-letter codes. Of those 64, only 61 code for amino acids. Now, one of them is AUG. This is the start codon. You've noticed in this slide here, and potentially this slide back here, AUG always encodes for our friend methionine. Now, methionine is what we call the start codon. Now, the remaining 3, which we have UAA, UAG, and UGA, as you will recall, are stop codons, meaning they terminate protein or polypeptide synthesis. So we have this handy dandy table. You've probably seen something similar to this, where we have the first position here on this column, I'm sorry, in this set of rows. We have the second position over here and the third position here over here. And so where we have the first position is U, the second position is U, the third position is U, we get U, U, U. And that corresponds to PHE, which is our friend phenylalanine. And so we can use little codon tables like this to specify what our amino acids are. And again, you'll notice that some of our amino acids have multiple triplet codons, right? Leucine has 4, proline has 4, threonine here has 4, but others have only 1, right? Tryptophan only has 1. right? Lysine and aspargic acid here only have two, and so on and so forth. So some have multiple, some have one, and there are a few that correspond to stops as well. Now, the genetic code is what we call unambiguous and degenerate, meaning that every codon has one meaning only, and it's unambiguous, meaning that when I have UUU, that only means phenylalanine and nothing else. Now, it is also what we call degenerate, meaning that many of the amino acids are specified by more than one codon, as we made mention to it. Now, you might ask yourself, why is it important that our amino acid code is degenerate? Well, the idea here is that when something is degenerate, a mutation can cause codon changes, but also no change to an amino acid. So, for instance, if we look at our friend serine here, if we have a mutation that changes it from UCU to UCC, well, we still have serine, right? Or if we went from UCU to UCA or UCG, again, we still have serine. So that's what we mean by degenerate code. But again, it's also unambiguous, meaning UCC can only be serine, and that is it. Now, there are some there's some sort of interesting things that we've noticed throughout the years. Well, I shouldn't say we've noticed, that scientists have noticed, I should say, is that there are, with a few exceptions, very little wiggle room in terms of the universality of this genetic code, meaning that the the genetic code you use to make serine or proline and so on and so forth is pretty much identical across the entire tree of life. Now, there are some examples where this doesn't hold up. Now, there are some mitochondria and a few bacteria where the genetic code does differ pretty dramatically. There's also an example here where we have AGA, which is actually a stop codon in mammals. But in some organisms, their codons are actually for what we call non-standard amino acids, meaning there's these funky amino acids that aren't, say, one of these 20 that we see. So there's lots and lots of examples of sort of funky things, but by and large, once this system evolved billions of years ago, it has changed very, very little over time. Very much the same way the genetic code, ATC, and take some wacky and wild secondary structures as well. So we'll talk about this idea of transcription by actually just looking at bacteria and the way they do it, because it's starkly similar between bacteria and us, which is kind of a good thing. Though ours is slightly more complex with a little bit different players, but the concepts still readily apply. So we have DNA, and it gives rise to RNA molecule. We call this what we call a transcription unit. And transcription itself is initiated when RNA polymerase, so an enzyme that just synthesizes RNA from DNA, binds to a promoter sequence, triggering a local unwinding of the double helix. So it does what DNA gyrase does when we talked about DNA replication. Then our friend RNA polymerase initiates the synthesis of RNA by using one DNA as a strand of template. That's going to be our template strand. Now, after RNA synthesis is initiated or transcription is initiated, the RNA polymerase itself moves along the DNA template, unwinding the helix. So it has its own helicase activity, so unwinding the helix or breaking it apart. Then the unbound DNA is called a transcription bubble. That should sound familiar, right? It should sound a lot like the replication bubble we discussed in the previous part of this lecture. And it's actually very, very similar. Different machinery, but similar concept. Now, eventually, once the transcription bubble is open, RNA polymerase will eventually, and things are done, RNA polymerase will eventually dissociate from the DNA template, leading to the termination of synthesis and the release of the RNA molecule. So let's take a peek at it. Right, our transcription unit is between the promoter and slightly past the terminator. Now, what happens here is that a binding of RNA polymerase, which causes a local unwinding of the DNA, so relaxing the double-stranded, I'm sorry, unwinding the twisted ladder. Then RNA polymerase initiates synthesis by first opening up the bubble, so replication bubble, transcription bubble. Then RNA polymerase moves in a 5' to 3' direction. So that should sound familiar. It acts the same way DNA polymerase does, 5' to 3'. So it moves in a 5' to 3' direction, using the old strand or the template strand of DNA as a template to synthesize a brand new piece of messenger RNA that has polarity very, very much like our um, our DNA itself. And as the RNA polymerase goes and goes and goes, it eventually hits the termination sequence, which then it falls off. The RNA polymerase is released to go back and do more transcription as necessary. But then we have a primary RNA transcript. So that's the overall um, flow here. Now, there are some important things that we need to discuss. Now, RNA polymerase, as we mentioned, and as you see here, needs a promoter. Now, what does a promoter means? Well, before we do that, we just need to talk a little bit about some terminology. Now, we have the term upstream and downstream, which essentially refer to the sequences located towards the 5' or 3' area. So when something is typically upstream, it's up here. So up always a purine and is usually almost always our friend adenine. Now, about 10 base pairs upstream from the start of the site is what we call the TATAT box, or simply just called the TATA box, which is what we call the -10 sequence, or what we call the PrimNow box. Either works. And we also have ATs at the -35 position, which is the sequence TTGACA, which we call the -35 sequences. Now, the cool thing is these are evolutionary conserved, meaning that TTGACA is the same across almost all species. So this promoter here and the start of transcription is going to be about here, right? But the RNA polymerase needs to recognize these two conserved mechanisms, these two conserved DNA sequences, both upstream from where we were. And so we can sort of look at this, right? We have the transcriptional start site here. That's where the first, that's the first base on our brand new piece of messenger RNA. But -10 and -35, you notice the TATAs and the TTGACAs. That's our promoter DNA sequence. That's what our friend RNA polymerase is reckoned. So is there a lot more to this? Yes, of course there is. But the reality is it's much more simple than DNA replication. Why? Well, there's only one enzyme. And RNA polymerase as an enzyme has multiple activities that it does, right? It has the activity of gyrase, relaxing the double helix. It also has a ligase activity. It also has helicase, meaning it can split DNA apart. So it does all of its stuff, right? Unlike DNA replication, there's multiple things the single polymerase can do. So this brings us to the final part of today, which is actually going to be translation. So what do we do when we make a messenger RNA? The first is what we do with that messenger RNA is translation. So we have a couple of characters in our cast we'll just briefly discuss. The first is ribosomes, which are the protein RNA hybrids that carry out the process of polypeptide or protein synthesis. Then we have tRNA molecules, which align the amino acids in the correct order. Now we have aminoacyl tRNA syntheses, which attach amino acids to their appropriate tRNA molecules. Then we have messenger RNA, which is what we just talked about. And then we finally have protein factors, which actually facilitate some aspect of translation. So those are our cast of characters. Now, our ribosomes are the site at which polypeptide or protein synthesis occurs. Now, they are made out of both ribosomal RNA and protein. Depending on the organism, they can be upwards of 80% rRNA, but usually it's about a 60-40 split between protein and RNA. But again, it does depend. Now, the ribosomes themselves in eukaryotes, as you recall, are either found on the endoplasmic reticulum or floating in the cytoplasm. Conversely, for bacteria and all other prokaryotes, they're always found in the cytoplasm. So let's look at some structure of our ribosomes. So there is a large upper part, which we call the large subunit, and a smaller lower part, which we call the small subunit. So easily and aptly named. Now, the small subunit is measured in S's. These are what we call Svalbard units. You don't really need to know what a Svalbard unit is, but just know 50S is larger than 30S, 60S is larger than 60S. That's all you need to know. And what you notice is that these large and small subunits are not comprised of just a single protein. Yeah. But you also notice that the small subunit has 16S rRNA and 18S rRNA down here as well. So uh, some slight differences here between our eukaryotes and prokaryotes, including an overall larger size. The average prokaryotic ribosome is 70S in size. Conversely, the average eukaryotic ribosome is about 80S in size. So similar structures, different types of proteins, different types of rRNAs, but overall the same standard structure: large subunit, small subunit. That's pretty much it. And this is just a handy dandy table, sort of going over some of those more specifics. Now, our friend the ribosomes have four distinct sites. So what we call the first, the mRNA binding site. We have the A site, which would be the amino acyl site, the P site or peptidyl site, and the E site and the exit site. So let's take a peek what those look like in 3D space. So we have the mRNA binding site down here, which is on the lower, I'm sorry, the small subunit of ribosomes. Then we have the EPA site, a combination of both the upper, I'm sorry, the the large and small uh, subunits of the ribosome. So that's what it looks like in a cartoon diagram, and this is what it looks like in more 3D actual space, where we have the 50S part of the ribosome here, the 30S, the E site, the A site. We have the exit channel up here, which we'll get to in a moment. We have the RNA, and we have sort of the overall thing here. And then we can look on the inside again. We have the exit channel here. Now you'll notice there's just like a bunch of beads coming out of here. All those beads mean are amino acids. So this is the exit site is where the protein that's being synthesized is exiting from the ribosome. But again, EPA, EPA, EPA. Keep that in mind. That directionality is important. EPA, EPA, EPA. Keep that in mind. Okay. So let's talk about how we carry amino acids to our ribosomes. And we actually carry these via tRNA. That shouldn't be new to you at this point. But tRNA is a type of RNA, and it has an adapter that binds both a specific amino acid as well as the messenger RNA sequence that specify the amino acids. Now, each tRNA is linked to its amino acid by an ester bond, and tRNAs are named for the amino acids attached to them. So, for example, if I have tRNA ALA, this means I have a tRNA that is charged, or we could call it has a alanine attached to it. So it's charged with alanine or has an alanine attached for it. Both ideas are similar. So let's look at our friend tRNA here, where we have an amino acid attachment site up here at the top, a bunch of RNA, some hydrogen bonds holding things together. Then we have this anticodon loop where we have a C, G, and then we have the wobble position. Um, we have the wobble position here. Um, and you'll notice that's our codon. And then it wraps back around here. So there's 3', 5', so on and so forth. Now, we have an entire class of amino acids that are, proteins that are called amino acyl transferases, which will actually charge or attach an amino acid to this amino acid attachment site. So, for instance, we have the amino acid alanine is added via an ester bond, AOC, that's an ester, very much like our DNA, which has phosphoester bonds. This is just an ester bond. That's how we attach the amino acid here. Um, and that's pretty much it. That's pretty much what we're looking at here for our friend here. Now, the important thing I'll make mention of before we move on is the anticodon. The anticodon is corresponding to the codon. So that's that triplet 3-letter code on our messenger RNA. So I just settled this, so we'll move on to the next slide. But we do have an important thing we need to make mention of. I made mention of this wobble position. It's an I. So what does the wobble position mean? Well, it actually refers back to the wobble hypothesis, which allows for some funky and unexpected base pair. Now, in mRNA and tRNA, and when they line up together on the ribosomes in a way that permits an overall level of what we call flexibility or wobble in the pairing between the third base of the codon and the corresponding base of the anticodon. Now, you'll sort of see what this looks like, where we have the anticodon here, and again, it's in this slight odd orientation where the third codon simply doesn't matter all that much. This is what we call the wobble hypothesis. So in the idea here, um, I'm actually going to skip this. We don't need to talk about inosine. But what we have here is we have the wobble position where we have UCU on our tRNA. But because of the sort of the funky orientation here, UCU can actually recognize UGA, but it can also recognize UGG. So the kind of the cool thing here is we can actually look at this table where we have the bases recognized in the codon, so the third position of the codon in the messenger RNA, and then the base in the anticodon. So we have A recognizes U, C recognizes G, U recognizes A or G recognizes C or U, and then we have a final base, which is inosine, which I skipped over a little bit. It doesn't really matter too much, but inosine can recognize U, C, or A. What you need to take away from this is not this table, but that third position is the wobble position, allows for and funky complementary base pair. Now, as I made mention of, tRNAs need to be charged. And when I say charged, we simply mean to attach a amino acid to our tRNA. And as I mentioned, this is carried out by a group of enzymes called aminoacyl tRNA synthesis. So we can look at what this looks like. We have an amino acid plus tRNA with a free hydroxyl group here. We consume ATP, producing AMP and 2 phosphate groups. And we use that energy to essentially tack on our amino acid here to our tRNA. And so that's what we have in terms of synthesis of tRNA. This again is also called charging of tRNA, hence what you're talking about. So we can look at a cartoon example of this here. We have aminoacyl tRNA synthase. We here. AMP binds to the amino acid. Then AMP is replaced by tRNA, creating that aminoacyl tRNA. And then the aminoacyl tRNA is then released from the enzyme, and the enzyme itself can keep doing what it needs to do. And so we can sort of look at this again in a more 3D model here. tRNA synthase, 3' stem, ATP, an amino acid, 3D structure, what this looks like. Okay, so how do we start translation? Well, we first start with the very first codon, the start codon, which is AUG, or methionine. So the mRNA actually has to be, in our eukaryotes, has to be first exported from the nucleus into the cytoplasm or to the Golgi, the endoplasmic reticulum. And that untranslated sequence at the 5' end has a start codon, which is the first to be translated, typically AUG. Now, at the untranslated region of the 3' area of our messenger that are simply untranslated, so they don't make it into the final protein. And we also see mRNAs that have 5' caps and 3' poly 8 tails, which are also not translated. So let's take a peek what we have here. We have prokaryotic mRNA. We have the start codon, the stop codon. This is the coding sequence. This is what creates our protein. And then we have the untranslated areas here and untranslated areas here. And we also have this interesting piece of bacteria, which we call the Shine-Dalgarno sequence. Say that 5 times fast, which actually helps them recognize the ribosome. Then we have on eukaryotes the same thing. We have the untranslated areas here, but in eukaryotes, you notice we have a 5' cap and a 3' poly 8 tail. So distinct things from our bacteria. But again, start, stop, coding sequence in between, that's the name of the game here. Now, eukaryotic mRNAs are what we call monocistronic. Now, most messenger RNAs in eukaryotes are monocistronic simply because they only encode just a single polypeptide. However, in bacteria and archaea, we also have many mRNAs, I should say, that are polycistronic, meaning that they encode several polypeptides and they have overall related function. So what that means is that I can have a single messenger RNA strand that encodes for actual 2 things at the same time. So let's get into the process of translation. So translation begins at the N terminus, so the start of the protein, and proceeds adding amino acids until we get to the C terminus at the very end. Now, the mRNA itself is read in the 5' to 3' direction, and we can divide translation into three distinct stages: initiation, elongation, and termination. So let's just take a peek at what these look like. So initiation, our components, think everything comes together. Our mRNA comes into the small, in between the small and the large subunit of the of our ribosomes. The ribosome forms around it. We have the very first amino acid. Um, we have our very first tRNA come into the P site. Now, once that happens, that's initiation. That's really all that's going on. Once that starts, we start to have elongation, where essentially during elongation, our amino acids are brought to the mRNA via tRNAs, and they're added one by one to a growing strand. And then we eventually have termination, where a stop codon is reached, and we have what's called a release factor that binds to the A site, causing essentially the falling of part of the ribosomes, allowing for the completed polypeptide to be ejected and then go do what it needs to do. So that's going to be the end of today's class. That's all we're going to go over. There's a lot more here, but we'll cover a little bit more about this in our subsequent class when we talk about control over transcription and translation. So I hope you guys learned a little bit of something. If you have any questions, please let me know. Otherwise, take care.